Hey there, we're going to talk about our meiosis notes from this activity from packet page 72. So we need to cut out the table, which I have already done, and I have glued it into my notebook. And we just need to go through and we need to label the different stages of meiosis. So meiosis 1 and meiosis 2. So starting up at the top, right, with meiosis 1, we always start with interphase, right? Just like in mitosis, meiosis begins with a diploid cell, meaning it has 2 copies of each chromosome. During interphase, the cell, right, it grows a little bit, it gets a little bit bigger, it copies the DNA, and it makes any necessary proteins and gathers any nutrients it needs before cell division is going to occur. After interphase, we have prophase 1. In prophase 1, we have our first event that increases genetic variation, right, which we call crossing over, right, where the copy of chromosome 1 and the original chromosome 1, right, they kind of exchange genetic information to increase genetic variation between the different gametes that it will create. After prophase 1, we have metaphase 1, right? And in metaphase 1, we have another event, independent assortment, where the homologous chromosomes, right, line up in the middle. Here, we're only showing 4 chromosomes. Again, humans, right, we have 46 chromosomes. I don't have enough time or space to draw all of that, so I'm just showing 4 chromosomes. But they are going to line up in the middle, right, with the homologous chromosomes lining up against each other. So, it'd be chromosome 1, 1, 2, 2, 3, 3, so on and so forth. But really the order, right, is not determined by anything. So the order in which they line up, right, it could be chromosome 7, 7, 5, 5, 1, 1, 12, 12. Right, so the order they line up is completely random. So we call that independent assortment. After metaphase 1 is anaphase 1, where the homologous chromosomes separate. So, right, this copy of chromosome 1 goes to the left, but this copy of chromosome 1 goes off to the right. So the homologous pairs have separated. And then we have telophase 1, right? A new little nucleus kind of starts to form. We start to get that pinching of the cytoplasm for cytokinesis. And then we actually have cytokinesis 1. So now we have 2 daughter cells, but we are not finished, right? We've got to do meiosis 2. So we basically start over with prophase, but now it's called prophase 2. There is no crossing over in prophase 2, right? That crossing over only happens in prophase 1. Prophase 2, right, we simply dissolve that nucleus that formed for a brief moment during cytokinesis 1, right? That nucleus starts to dissolve again. And then after prophase 2, we have metaphase 2. Now the chromosome is lining up in the middle, kind of just like it does in mitosis. Meiosis 2 and mitosis are very similar with what's happening to the chromosomes. So they're lining up in the middle, and then in anaphase. 2, the sister chromatids are pulled apart, right? So here we had that double chromosome, but now the sister chromatids have been pulled apart, moving to opposite ends of the cell. And then telophase. 2, the nucleus starts to reform. We start to get that pinching of the cytoplasm. And then finally, we have cytokinesis 2. And really, right, this cell would be right here. And then we should have really another cell represented for this one, because at the end of cytokinesis 2, right, we should end with 4 haploid daughter cells. Right, in the end, or sorry, at the very beginning in prophase, right, we had 1, 2, 3, 4 chromosomes because we were diploid. But here at the end, with our daughter cells, the number of chromosomes we have is only 2. Right, this cell has 2 chromosomes. This cell has 2 chromosomes, 2 chromosomes, 2 chromosomes. So we went from having 4 to 2. So we truly have cut the number of chromosomes in half to create 4 haploid daughter cells. And if this is a male, these will become sperm. If this is a female, one of them will become an egg, and the other 3 will become what we call polar bodies, and any genetic information in them will be basically thrown away. Only one of the cells will become an actual egg that can undergo fertilization to be used in sexual reproduction. Unlike males who would create 4 viable sperm. So that is it for our notes here on meiosis. If you have any other questions or concerns about meiosis or this activity specifically, please let me know.